Hi, everyone. We hope that we have gathered a number of experts in fish vaccination that will give you an overview of what's been going on in the past years concerning fish vaccination. And I'm going to start by giving you a talk on the adaptive immune system, which is, of course, the basis of vaccination. Okay, so as a start, well, just a brief overview. Of course, in aquaculture, what we have is intensive culture, and the fish are in really high densities. As a result, we have stress, accumulation of organic material, and low oxygen levels. And this provokes, of course, that fish are much more susceptible to pathogens, and also that the pathogens, of course, impact the fish higher. So the thing is that we can have different kinds of pathogens, bacteria, viruses, parasites, fungi. These are some examples of the pathogens that we get, for example, in rainbow trout. So the thing is that, of course, vaccination is the most efficient way to control these pathogens, but this is not always possible. So for example, in the case of bacteria, where we could use antibiotics, but of course they are limited, their use is limited because of all the implications this has. There are some intraperitoneal vaccines that are effective for some pathogens, but still there are many pathogens for which we don't have these vaccines. And of course, we would also aim to get some vaccines that are administered to the fish in a more efficient way, like, for example, oral vaccines. In the case of parasites, again, we have the possibility of using some treatments, but their use is also limited, and there are right now no commercial vaccines against any parasite. And in the case of viruses, we don't have really authorized treatments when a virus appears. And there are a few commercial vaccines available, but usually their effectiveness is not that great. So with a few exceptions, of course. So when we want to develop a vaccine for fish, the first thing, of course, that we have to do is to identify a protective and immunogenic antigen. We have to think of the administration route. Depending on this route, we also have the possibility of doing different regimes or different boosters. We can also include or not some adjuvants, and I would talk about this later today, also about the adjuvants. And of course, vaccination should occur before the pathogen exposure, give time for immunity to develop, and this in fish does take some time. And also, of course, take some other factors that can influence this, like water temperature, size of the fish, and things like that. So what are the challenges specifically for fish vaccines compared to other animals? The thing is that we, of course, need to understand how the adaptive immune system works. And as we learn more and more, we realize that it's not like we always thought that it would be exactly the same as mammalian immunology, but there are some really big differences, especially in adaptive immunity, of course, and that will condition how everything works. Of course, we also have to take into account that different fish species may react differently. We have to take into account that fish are really diverse group, and actually they have really important differences in their immune system from one species to another. Also, that we need to vaccinate in many cases in really early developmental stages. So it's important to know when adaptive immunity starts. Of course, as we said, let's say what we all want would be to have oral vaccination strategies in which you could just administer the vaccine in the feed. And for that, we really have to take into account tolerance mechanisms, because in the gut, the immune system is really restricted so that animals are not continuously responding to oral antigens or to commensal bacteria. So this is something that we really have to bridge if we want to do oral vaccination. And we have to identify, of course, specific adjuvants for each case with low side effects, if possible. And another important issue is the that we always need to come up with tools to monitor vaccine efficacy and find correlates of protection so that we don't have to do challenges with a lot of fish, especially now with animal welfare and things like that, so that we don't have to do challenges every time we want to check if a vaccine works. So with all that, of course, what we infer is that it's really important to understand how the immune system works, and especially adaptive immunity. So fish are actually the first group of animals in which we find this acquired immunity that can respond in a second wave more specifically to an antigen. And that's the basis, of course, of vaccination. So this is just a small diagram of the innate immunity and the acquired immune system, in which we have mostly B and T cells. Okay, so I'm going to go a little bit through what we know about B cells and T cells. Of course, we work on B cells, so I mean, it's not going to be that balanced. It's going to be more towards B cells than T cells, but I also will have a brief thing on T cells as well, because this is really what happens when we inject the vaccine. So you have cells of the innate adaptive system that take up the antigen, and then they act as presenters, and they present the antigen to T cells, and then T cells, on the other hand, help B cells. But this is not, you will see that this is not exactly maybe what happens in fish, as B cells, for example, might also be implicated in the first stages of vaccine recognition, in addition to being afterwards activated as memory or plasma cells. Okay, so well, normally what we get when we inject something to an organism that has adaptive immunity, you get like a primary immune response. And when you get a second exposure, you get a secondary immune response. And that's the basis of vaccination. And this secondary response, of course, is normally more prolonged and more extensive. And of course, due to the fact that in the primary immune response, we have developed some memory cells, and we have this response for both T cells and B cells. Okay? So then for B cells, what B cells produce really is immunoglobulins that, when are secreted, is when we refer to them as antibodies. And antibodies are made up of a pair of heavy chains and a pair of light chains, which each of them have a constant region and a variable region. So this is like an schematic overview, and here on top is the antigen binding site, and it's the variable region. And this is really what allows the immune system to be able to recognize a wide selection of antigens, because in cells of the adaptive immune system, what we have is that there is a recombination process in which different fragments are selected in each B cell, so that we end up with one cell producing a specific type of antibody that is unique or more or less unique. And that way, each B cell recognizes a different antigen, and we have a pool of these different cells, and then one of them is selected when an antigen appears, okay? So in mammals, we have 5 different types of antibodies according to the heavy chain of the immunoglobulin. So we have IgA, IgD, IgE, IgG, or IgM. And this, you will see that it's not exactly the same in fish. So the constant region of the immunoglobulin determines how the antibody is secreting, how it will get distributed through the body, and its activity. But it doesn't condition the antibody specificity. This is the variable region. So then the variable region is both on light chains and heavy chains, and contains the antigen binding site, so that due to this recognition, actually what we have is that, for example, in adult humans, we have 100 million different specific antibodies that can recognize, of course, a wide range of things. And then when you expose the individuals to an antigen, then a pool of these cells will be selected and they will start proliferating, and this is what they will create an immune response. So these are just the classes of antibodies and with the different functions. So at the end of the day, what we have normally in mammals is that we have a primary response in which we have a first wave of IgM and a second small wave of IgG. And then when the individuals are exposed the second time to the antigen, we have a little wave of IgM and a huge wave of IgG that it's faster and stronger. Okay, so this is of course what we aim for, but we don't have IgG, you will see. So this is different in fish. Okay, this is just of course that we have to take into account that this takes time to develop, so we have to do this in advance, that throughout this process cells differentiate into plasma cells, and there is like a gradual rise of antibodies, and it takes like 1 or 2 weeks. Okay. And in the secondary response, in some cases, for example, in humans, these memory cells can survive for up to 20 years in specialized niches, and then you can activate them. And of course, these plasma cells secrete larger quantities of antibody, and the response is much faster. Okay. But then I'm going to go through a brief overview of how this differentiation process, or what happens to B cells in mammals when they see an antigen, and then compare to the situation in fish. Okay. In mammals, what we have is that when a B cells that we call B2 or conventional B cells encounter an antigen, what they do is that they organize themselves in a germinal center, okay? And that they are produced in the follicles of secondary immune organs, which in mammals are the spleen and the lymph nodes, okay? So in this case, what they do is that these B cells, in close cooperation with T cells and dendritic cells, they become activated and they start the differentiation process, it's called. So throughout this differentiation, they come out either as plasma cells or as memory B cells. So then what happens through this differentiation process? What happens is that when a naive B cell starts, they have IgM and IgD on the surface of the cell, and this with the same antigen specificity for both immunoglobulins. And then when they are activated, they lose IgD, and then they start differentiating either to plasma cells or memory B cells. So in some cases, they keep producing IgM, but in most of other cases, in mammals in the germinal center, what happens is that this heavy chain, this constant region of the heavy chain of the IgM is replaced by either IgG or IgA or IgE. And this depends on the antigen that has originated the response. Of course, for example, IgA in humans is mostly associated with mucosal immunity, and IgE, for example, with responses to parasites or allergies, things like that. So then, well, I didn't mention that this change of heavy chain and this, it's called class switch recombination. Okay, so this is what happens in the germinal center. And then another thing that happens in the germinal center on top of that is that these variable regions of the antibody start to get mutated. So you start variating a bit some of these regions, and at the end there is a process by which those that have higher affinity for the antigen are selected, whereas those that have lower affinity, they just die. And that's a way also to increase the affinity of the antibodies. And this is due through a process that is called somatic hypermutation. It's the process in which the variable region gets mutated and the ones that have the higher affinity are selected. Okay? So then what happens, of course, during this process when a B cell is transformed into a plasma cell, in addition to losing the surface IgD, of course, there are lots of changes in the cell. And we know that there are some, for example, specific transcription factors that they change from what happens in a B cell to what happens in a plasma cell. So, for example, you will hear me later talk about BLIMP1, which is like a really well-known factor for plasma cells, or IRF, for example. This is what happens in mammals, and this also happens in fish. So then, of course, one of the other things just to mention that we thought happened was that in mammals, it was always thought that when B cells differentiated to IgG plasma cells, they lost surface immunoglobulin production. But now it has seen in humans even that this is not exactly true for plasma cells of other isotypes. So, for example, IgM plasma cells still retain IgM on the surface, and IgA plasmablast as well, or plasma cells. And then the other thing that we have to take into account is that this is like the regular situation in mammals, and what happens when we think of a B cell response in mammals. But actually, there are many other types of B cells that do not exactly the same thing. So we have what we've talked about would be what we call follicular B cells. And then we have other types of B cells that can consider even elements of the innate immune system, which are mostly the B1 cells and also marginal zone cells in the spleen. But B1 cells, what they mostly do is that they are responsible for the production of natural antibodies. So you have to take into account that what I've told you about the germinal center does take a while to develop, even in humans or in mammals. So you need something to block the pathogen while this response is being mounted. And these are B1 cells. So they produce antibodies that are not as selected and are not as with that high affinity, but they are able to block somehow a bit pathogen. So they do not require T cell help, and they sometimes are also well known because they produce lots of amounts of interleukin-10. So actually, these B1 cells are really important in some specific pathologies, even in humans. For example, they have been shown to be really important in case of encapsulated bacteria and things like that. So they do secrete like spontaneously lots of amounts of IgM. They are localized mainly in the peritoneum or on mucosal surfaces to face the pathogens as they come. They never switch, so they are always IgM. There is no or very little somatic hypermutation. They do not require T cell help. They self-renew, and they can develop a bit of memory, but of course not in the same way as B2 cells do, of course. And they are mostly induced by what we call T-independent antigens, which are, for example, LPS or repetitive modes, repetitive structures in pathogens, which activate the cells, like in a first wave, like it was an innate cell, and they start producing lots of IgMs, okay? So at the end, the situation in mammals is you have both things, okay? You have within the germinal center, you have this response really specific, but that takes a lot of time or more time to develop. And you also have outside the follicle, like extrafollicularly, some B cells, mostly B1 cells, that become activated, and they produce short-lived plasma cells or plasmablasts, and short-lived memory responses, but that are faster, okay? So then what happens in fish compared to all this? We have to take into account that fish evolutionary are the first that have most or almost all the elements of the adaptive immune system, but of course there are some differences. So there are no lymph nodes, There is no bone marrow, so the head kidney is the main hematopoietic tissue and also the place where B cells mature. Of course, all these conditions, what happens? We do have a spleen, but then, of course, the main difference is then the immunoglobulins that we can express. So here we have the immunoglobulins that I talked to you about in mammals, IgM, IgD, IgE, G, and A. But in fish, we only have, for a while, we thought that we only have IgM and IgD. And then we have IgT, which is a teleosed fish-specific immunoglobulin. I'm going to talk about it. Okay, so the thing is that in fish B cells, we don't have class switch because there are no germinal center or not as they are in mammals. Some groups are starting to see that there are some groups of cells that get formed when you get an immune response or sometimes when you get a repetitive immune response. But of course, these structures are not as organized as a mammalian germinal center. So in consequence, of course, there is also low affinity maturation. And what the consequences of this when we talk about vaccines is that sometimes what we see in fish is that there is very little, in some cases, correlation between antibodies and protection. The secondary response is not as high, and sometimes it's not even possible to see it, depending on what. And also what we see is that for bacteria, things seem to be different, but for viruses, which are not as immunogenic, of course, the immune response is really low, and viral vaccines have low effectivity, as let's say classical viral vaccines. So the thing is that, for example, we do think that really what we have to think about is that fish B cells resemble maybe more these extrafollicular B1 responses than actually what you get in a B2 conventional mammalian B2 situation. So it's important to understand how the different fish B cells are activated. And of course, it's important to generate new tools to study B cells in fish. So what subsets do we know that exist in fish? So we know that naive B cells in fish, like in mammals, they co-express IgD and IgM. That we know. And we know, and we'll see that when these cells are activated, they lose surface IgD. This also we have verified, and this is actually one of the ways in which we can differentiate between naive B cells and activated B cells. If you have for your species an antibody against IgM and IgD, you can play with that and differentiate naive cells from plasmablast plasma cells. And then we know that I will tell you later about this, that in fish we have, well, also in mammals, but in fish, we can see a subset of cells that exclusively expresses IgD. And we don't know yet how this differentiation occurs. We just know that we see these cells. And then the thing is that we have this other immunoglobulin, IgT, which is not actually a switch of one antibody to the other, but it's an independent lineage from the start. And this independent lineage has been shown, well, I will talk about that later, but we really still don't know how this differentiation occurs in IgT cells as well. Okay. So I'm going to talk about what we know of fish IgM cells and things that we have been doing. So one of the first things that is important to take into account is that some years ago, the group of Oriol Sunyer in the US, they discovered that B cells in fish could be phagocytic. And this, of course, was an important thing, because then they also realized that B1 cells from mammals were also phagocytic. So this not only has consequences in the way that fish B cells sense the antigen and how they can present it, because you have to take into account that B cells are also antigen presenting cells. that have MHC class 2 on the surface, so they take antigens. So in mammals, they only take the antigen through the immunoglobulin, through the BCRs, the B cell receptor. But here, being phagocytic, of course, this implies that they can present things in a different way. But it also, they shown with other works that also this phagocytic activity is a way to contribute to pathogen clearance. And they do produce antimicrobial peptides, and they do act a bit like a macrophage would do, like an innate, having lots of innate receptors that can allow them to sense the pathogen. So this is, for example, the work we did in which we found that B cells from different sources express lots and lots of TLR, which are the receptors for innate patterns in pathogens, okay? And of course, this capacity that they have to detect antigens and to react also conditions the way they behave. For example, this is one of the things that we did in collaboration with Niels Orensen, that we saw that when you inject a DNA vaccine into the muscle of fish, one of the main cells that get attracted there in an early time points are B cells. And this is something that would never happen in a mammal, for example, where B cells never go to a solid tissue. And we even optimize the method to get these cells out of the muscle. And you can see here that we have IgM cells, also IgT, and even a bit of T cells, but the main thing was having B cells there as an early start. So then a similar thing, for example, happens when you inject something in the peritoneal cavity. So what we saw is that, well, for a long time, people had been focused on the influx of macrophages that you get in the peritoneal cavity when you inject something. But we realized by flow cytometry that you have these small cells, lymphocyte cells, that also increased a lot in the peritoneal cavity in the early stages of an antigen exposure. We injected either LPS or E. coli or VHSV. So you don't only see an influx of myeloid cells, but also of lymphoid cells. And actually, when we analyze what these lymphoid cells were, you will see in blue are the IgM positive B cells. And you will see here that while in control fish, you have around 20-25% of B cells in the peritoneal cavity, when you inject things like E. coli or LPS, you can get up to almost 50% of the cells in the peritoneal cavity being B cells. So of course, this means, again, that B cells are responding to inflammation, to early moments of inflammation, as a B1 cell would. And then, well, we did this other work in which we compared actually B cells from mice to B cells from trout. And really, this is just a brief summary, but we came up with different things in which B1 cells from mice were similar to trout. Like, for example, the way they respond to BCR cross-linking is similar. And then, of course, I told you that it's also important to generate tools that can help us monitor B cell responses. So we have this IgM, IgD that you could do, but of course you don't always have an anti-IgD antibody. So we came up with this other strategy in which through flow cytometry, we monitored the size of B cells and the amount of endoplasmic reticulum that they have. So you have these kits in which you can monitor the endoplasmic reticulum. And as you see here, you have mostly in control conditions, the cells are smaller and have low amounts of endoplasmic reticulum. And then when you activate them, they start getting bigger and with more endoplasmic reticulum. And then this, of course, is just quantifying it. And if we sort out these populations, you can see here that the naive cells are round with a really large nuclei. And then the cells start to getting bigger and more cytoplasm. And the cytoplasm is full of endoplasmic reticulum, of course, because you have to think that these cells their main function is to produce lots and lots of amounts of immunoglobulins. So that's what they do. So then through this methodology, we could come up with sorted cells, and we did a transcriptomic analysis, and then we could really verify which markers are being induced, what transcription factors are induced specifically in these cells that are plasma-like cells. So for example, we saw this is one of the genes that codes for BLIMP1, the one I told you about, because, well, we have seen that rainbow trout expresses 4 different homologs of this gene. And in some situations, one of them come up. In some situations, all of them come up. But it's a bit. This is something that happens a lot in rainbow trout and salmonids, that many immune genes are duplicated or even many more times expressed. Another thing that we saw that also happens in humans, that it was really upregulated in the plasma cells, is BCMA. BCMA is one of the receptors of PAF, a cytokine that regulates B cell function. So BCMA was also really high upregulated in plasma cells, and IRF4 as well. So we have here like a tool to monitor. And then, of course, what we did is that we sorted the cells, we put them in culture, and we took the supernatants, And we did an ELISA to quantify the amount of secreted IgM, and we could verify that, in fact, these cells with a high reticulum and large size that expressed all these markers were really secreting lots of immunoglobulin more than the other ones. So you see, we have to find in fish other ways to monitor B cell activity, that creative ways. Okay, so this, of course, also allowed us to go further into the function of these cells, and this is also something that I wanted to highlight because this is not also common. Normally in mammals, when a B cell differentiates to a plasma cell, they lose MHC class II and they lose the capacity to present antigens. But here what we see is that this doesn't happen very often. So in most cases, when a B cell differentiates, they keep producing MHC class II. So it's like they're not fully devoted to only producing antibodies, but they also keep some of the early functions. And this is also the way in which we monitor the processing of an antigen. So this is the glucosane, and when it breaks down, it emits fluorescence, and we see that these cells still have the capacity to process antigen as well. And then fish IgT cells. Okay, so as I told you, this is a complete different lineage of cells. So normally B cells that only express IgT do not express IgM or do not express IgD. And one of the first works in which, after an antibody was developed against these cells, what they saw is that whereas in central immune tissues, the amount of IgT cells is really low compared to the amount of IgM cells, in here was this first work was in the intestine, there the number of IgT cells was higher than the amount of IgM cells. And what they also saw was that in response, not only in the intestine, but also in the skin and later on in the gills, was that in response to this parasite, here it's IC in these two studies, what they saw is that in this case, IgM responses were not as affected, whereas IgT responses were more responsive in mucosal surface to the parasite. And this happened both in skin and in gills. And through that, they heard that really IgT was devoted to IgM, and that was similar to IgA, which of course is different because in this case, it's not a class switch, it's a completely independent lineage of cells that is doing something else, so it's not exactly the same. And what we have also seen in some other works and some other people have seen is that you also can get IgT responses outside of mucosal surfaces, and these are really important. So we still are far from knowing what IgT is doing. So for example, here you see in response to a VHS infection, intraperitoneal infection, you see that in the spleen, well, this is a way to really show that some clones of cells are being expanded. And you see that IgT cell gets expanded in the spleen in response to VHS. So it's not exactly. And this is another example in which we did this work in which we analyzed the response to a parasite in the kidney in response to PKD, proliferative kidney disease, which is caused by a parasite. And we analyzed the three immunoglobulins, and actually IgT was the one that responded more to the parasite in the kidney. So again, we still don't really know what IgT is doing. And then, as I told you, we have these IgD cells, which it's a really strange thing. It's not that common, but it happens also in mammals, for example. So as I said, normally what you have is that you have cells co-expressing IgD and IgM, and normally they lose this IgD and then they become plasma cells. But by some process that is still not perfectly known, some cells, what they lose is the surface IgD, and they become IgD only. So in humans, these cells are mainly found in the upper respiratory tract. You can also see some of them in the blood, and they have like a semi-differentiated state towards plasmablast, okay? So they have some immune markers of plasma cells, but not all of them. They do seem to think that it's some sort of class switch recombination, that is dependent on the enzyme that is involved in class switch recombination. And they have been seen that IgD codes pathogens and commensals in the upper respiratory tract. And somehow the secreted IgD interacts with basophils. And although in humans they have not really detected these IgD-only cells in the intestine, it has also been seen that the intestinal microbiota is also coated with IgD to some extent, and that IgD seems to have some relation with food allergies, okay? So what we found in fish was, well, there had been this previous work in catfish a long time ago, in which they did detect these IgD-only cells. In the case of catfish, it was in the blood. But what we saw is that in rainbow trout, although in the spleen, what you get mostly are double positive IgM IgD cells, in the case of mucosas like gills or gut, you did get a few cells that were IgD only. And actually, in the case of the gut, if we exclude, of course, from this analysis IgT cells, we just focus on cells of what we call the MD lineage, then you see that really IgD cells make up for a lot of cells. So then the next thing we saw was what happens to the intestinal microbiota. And we saw that some bacteria were coated with IgM, but some bacteria were also coated with IgD. So as happens in mammals, somehow IgD is also contributing to mark these commensals. And another thing that we did was we suppressed completely, well, not completely, but almost completely, let's say, the microbiota of the fish by an antibiotic treatment, an oral treatment for several days with different kinds of antibiotics. And what we saw is that not only IgM gets affected, but also the transcription of both secreted and membrane IgD gets affected by this antibiotic treatment, meaning that somehow there is like a two-way interaction between IgD and the intestinal microbiota. And another thing that we can do to analyze B-cell responses, which is a bit, I hope I can explain it clearly, is that we can analyze through sequencing the repertoire of different variable regions that we have in a pool of B-cells, okay? So as I told you, what you have in a B-cell in the DNA, in the germline, is that you have these different fragments of B, D, and J. And then when a B-cell matures, they select one of the Bs, one of the Ds, one of the Js, and then they also go through mutation rates. So at the end of the day, you have a B cell or a group of B cells that has an exclusive segment here, okay? So we can take all the B cells in a pool, and we can amplify these sections through what we do is that we have primers either on the constant region of the IgM or the IgD or the IgT, and lots of primers in the different B regions. We pull all these PCRs together and we do a massive sequencing, and we get like all the different sequences that you have in a B cell pool. And then one of the first things that we saw was that the number of different sequences that you find in this pool is much higher, for example, in the case of IgM, in the spleen than in mucosal tissues. So what does this mean? This means that when you get a differentiation process, some of these cells will start producing huge amounts of immunoglobulins. So of course, they make more copies of one single immunoglobulin. So then the variability is reduced, okay? So this means that here we have more naive cells and here we have more plasma cells. But this not only happens for IgM, it also happens for IgD. And that was one of the things we wanted to see. And of course, it also happens for IgT, but actually the difference is not that far. Another thing that we can see through this technology is that we can analyze the CDR3 region. It's a region within this variable segment that really interacts with the antigen. And normally you have in a pool of B cells that are naive, you have CDR3 regions with different lengths, okay? So normally the distribution of lengths is like a Gaussian distribution. And when you have a clonal expansion of some B cells, what happens is that one size gets selected, of course. So then this Gaussian distribution starts looking a bit more like this, okay? So then what we saw for IgD as well was that in the spleen, you have a Gaussian distribution, but then in the gills and especially in the gut, this does not happen. So this means again that somehow IgD is being selected in the gut. And another way of looking at it is that we can really classify these sequences in sequences that are repeated 1 to 5 times or sequences that are repeated more than 100 copies. So we can consider that these cells are naive cells, and whenever we find the sequence repeated more than 100 times, we assume this is a sequence that comes from a plasma cell, okay? So what we see, again, is that in mucosal surfaces, for IgM, of course, the number of sequences that are repeated even more than 500 times is higher, and the ones for IgD as well. So again, this means that some IgD sequences are being selected and expanded like if it was a plasma cell, okay? And this also happens for IgD. Then with time, of course, we have been able to see these cells in gills and skin, and we can also see here by confocal microscopy that you have here, like double positive, and the IgD only and the IgM only that we have in the surface. Okay, so what happens normally in a fish when we have B cell responses? So you have to take into account that when a pathogen comes, what we have is that we have a first wave of B cell responses in mucosal surfaces that we have, in this case, all of the different types of B cells that we can find, the M's, the D's, and the T's. And then what we know is that there's a response in the spleen, mainly in this case, IgM, but we also have some IgT. And another thing that we have seen is that at later stages, these IgM cells or these specific IgM responses go to the head kidney, which is the equivalent of the bone marrow. And we think that there is where the plasma cells are stored for, we still don't know for how much time, but for some time, okay? So we have to, of course, take all this into account, but I hope I convinced you a bit that really B cell responses are not similar, not that similar to how B cell responses are in mammals. And of course, this conditions everything when we come to vaccines, okay? Then just a little bit on T cells. T cells, what we have is that we have two different types of different types of T cells. We have helper T cells that express CD4 on the surface, and cytotoxic cells that express CD8 on the surface. Okay, so the main function of CD4 cells is to help in the antigen, in the first stages of the antigen recognition, and then stimulate B cells, and also produce cytokines that can stimulate other cell types, such as, for example, also CD8. And then the CD8 become effector, and they can kill infected infected cells, sorry. And you have, at the end of the day, again, memory T cells, which is, of course, what conditions that they can respond later. But there are not that many works that have been focused. I think what we know about T cell responses in fish is much less than what we know about B cells, for example. So we know that fish are capable of killing infected cells in a specific way, and that we have CD8, and some cells express CD8, and some cells also express perforin and granulosin, which are the main enzymes through which the infected cell gets killed. Okay, so this is an early work from Trout, in which they develop an antibody against CD8, and they could sort out the CD8 cells, and then they could see that, for example, when they stimulated with PHA, these cells increased, and they started producing more perforin, more nulosin, more interferon gamma, more interleukin 4. And another thing that's important is that, of course, you get lots of T cells in the thymus, but also it's kind of different that in fish you have lots of T cells, CD8 T cells in mucosal surfaces, such as the gills or the intestine. And then in the case of vaccines, for example, with DNA vaccines, what they have seen is that you are capable also of inducing these cytotoxic responses. So actually what you see here is that fish vaccinated with a VHS vaccine compared to fish vaccinated with the empty plasmid, they can, afterwards, the cells from this fish can kill specifically cells that are infected with VHS and not cells that are infected with another virus. So somehow they are able to recognize the virus and kill specifically the cells that are infected with VHS. Okay, and then here what they did, well, is to compare also different cell lines, because one was matched with the fish in MHC, which is important for cytotoxic cells, whereas the other one was not. And concerning T cells subsets, this is normally what is known that happens in humans, mammals, that you have a T helper cell, and then depending on the antigen and depending on the situation, then and on the cytokines that are produced, these cells differentiate to what have been called Th1, Th2, or I mean, right now there are these subsets keep coming up, and there are more subsets now. You have also Th22 and things like that, but these are the main ones. So Th1 and Th2 were the first one. So Th1 is more, these T cell helper cells are more focused on cell-mediated immunity, whereas Th2 help more antibody-mediated immune responses. And of course, once they differentiate to Th1 or Th2, they start producing also a different pool of cytokines, okay? So then in fish, this was a work done in 2016 in trout. Well, they developed also antibodies, but in this case in trout, again, we have two different CD4s. So this is also something that should be taken into account for many immune genes. And they developed antibodies against both, and they could see that you had cells that expressed the two CD4s or cells that expressed only one or the other. So it's still not well known what each of these populations is differently doing, but what they could see is that when they stimulated with a pathogen, these cells responded in the way that you expected them to, producing interferon gamma and interleukin 17, interleukin 2, some different T cell specific cytokines. And this is another work also in zebrafish, in which they also develop an antibody against CD4. And they could see that these cells, they could sort them out and also see them that they co-express SAP70, which is a factor well known for T cells. And again, they could sort them out and see that they were producing all of these different cytokines. But of course, we still don't know what we don't know if this in fish, these different subsets are in fact different subsets. And of course, this is also something that in mammals is also not well defined, whether there are stages of differentiation or whether they are in fact completely different cells. But again, we do see that these cytokines that are particular of some subsets are produced. So it does seem that we have a more or less similar response. Okay. So just to conclude, like the take-home messages is that B cell responses in fish are quite different from that of mammals, and of course this will condition the response to vaccine. Also, there is a great lack of information regarding how T cells work in fish and whether T cell memory is achievable. There have not been that many works concerning T cell memory. And of course, effective vaccination is achievable, but we need to really understand how the adaptive immune system works to really individually optimize vaccines for each pathogen and its host. Okay, thank you.